start. Hello, everybody. Anything that you want to ask or to tell about the course before we start? Okay, so today I'm very happy to have here Jean-Yves Tinevez. Did I say that good? Who was trained as an experimentalist physicist, and he wanted to be a teacher, but he was too young for teaching, so he decided to go to grad school, and he did a master's in biophysics and then a PhD while looking at the cells at the inner ear, hair cells at the inner ear. And after that, a PhD. So from instead of a science teacher or physics teacher, he got his PhD in Dresden, in Germany. And, in Institute Curie. And then he moved to Germany, to Dresden, to do a postdoc on understanding how cells generate forces. And then he made a big transition, moving into a research engineer job, where you could be involved in many projects, helping many independent research projects get their images, cell images, and data analyzed. He did that at Dresden, then he moved back to Paris, and from 2017 he's leading the image analysis hub in the Institut Pasteur. Jean-Yves is the person who developed or led at least the development of TrackMate. Some of you know that software, at least the people in my lab, and many other projects. And he's a leader in bioimage analysis. I learned to know him from that venue and bioimage analysis conferences. So yeah, so go ahead. And if you didn't hear, then please interfere with the questions. Don't hesitate. We know that we are missing the background, right? With the terminology, and feel free to just stop and ask, because it's hard to know what we know exactly and what we don't.
Well, thank you so much, Assaf. I'm really delighted to be there. I've been to Israel once in my lifetime, a couple of years ago, and I sorely miss not being there. I see from Alan Zilster camera that the weather and the scenery is absolutely beautiful in Israel. Well, the only thing you get from me is my bedroom. Assaf actually kindly invited me for this lecture. So it's going to be a lecture about bioimage analysis and what makes it fit. If I understand correctly, you guys are computer scientists by background, by trade, right? Am I right? You can nod or actually say yes. Okay, right. So I'm a microscopist. Like if I had to be defined like this, I'm a microscopist. I spend a lot of time imaging stuff on the microscopes and then doing quantitative analysis of that. And we thought with Assaf that it would be cool to have this lecture to actually transition and see bioimage analysis as some kind of special part of data science and introduce it like this. So I'm going to warn you, the things I will present now are very, very basic. Like, it's initially adapted from the first part is adapted for lectures to biologists that have zero experience in imaging at all. So if you feel I'm going too slow, don't hesitate. And also, as we discussed, it's at least the beginning is going to be interactive. So I'll be asking questions, and I spend most of my days, just talking to in Zoom, talking to a cameras. I would, with zero interaction with humans, I would very much like if you could, just try and answer the questions you best. So roughly with Assaf, we thought, this would be a good schedule, good outline for the lectures. Basically, trying to see, what is bioimage analysis? What's kind of special for it, about it related to data science? Some kind of challenges if you go there as a career. What I think are some of the challenges you will face. I need to speak, because this is a lecture about data analysis, so I need to speak about data manipulation and scientific fraud. So the third part is going to be about that. Assaf also asked me about to speak about what's the career. So obviously I'm working in the Institut Pasteur, which is a an institute dedicated to research in biology. And so when you will interact with biologists doing image analysis, what's the different levels? And then finally, very kindly, Assaf offered me to actually give opinion about what I think is important. I just picked two subjects, but they're very, very, very subjective and maybe not relevant. And maybe a last part just to discuss with you. I will make a break in the beginning. For health reason, I cannot speak very loud, so I hope the sound is okay. Please let me know. Yes, no? Okay, good. Okay, so let's start. So the first part is about bioimage analysis. What's special about it if you come from data science? Can you guys tell me what this is?
Pipe.
A pipe. Everybody agrees?
Ce n'est pas une pipe.
Ce n'est pas une pipe. Yeah, actually, this is not a pipe. This is an excerpt from a famous painting from René Magritte. Maybe you know that he's a surrealist painter of the beginning of the 20th century, I think. And he was fascinating by the relationships between what we see and how in imagery and how we vocalize and how we think about it. And the painting is actually called the Treachery of Image. Treachery, it's a big word. And for René Matisse, he was presenting this painting, quickly became famous. He say, Sir Magritte, you say this is not a pipe, but obviously this is not a pipe. And he would say, no, this is not a pipe. It's a spatial arrangement of colored pigments. Right? Despite the way our visual cortex, our brain works is increasingly powerful, right? Just from this, actually, I think it immediately identified a pipe from this spatial arrangement of colored pigments. And you can actually go much beyond that. If I ask you, is the pipe lit, yes or no?
What, sorry?
Is the pipe lit? Is there tobacco burning in it?
No.
No, right? Okay.
Maybe.
Vas-y? Yes, sorry?
Maybe.
No, I think you would see tobacco and maybe undulating fumes about that. And the mouse pipe, is it facing the left or the right?
Right.
Obviously, right? So you did image analysis, and image analysis is about that. You have like a spatial arrangement of things, and you want to make sense about it, like trying to guess that. But okay, so that's a small, let's say, divagation from the subject. What's kind of special for bioimage analysis is that it's really related to images, and we, despite being mammals, we're equipped with a visual cortex which is incredibly powerful. And it's so important that in our lives, that the brain takes shortcuts which are incredible. Like we confuse the representation of a pipe with the pipe itself and the object itself. And it's incredibly powerful, but also dangerous for our science, because it's all about quantification and scientific data. So I have a chance to come back to what you just did, right? First, the first question is, how is it possible that you identified a pipe that quickly? It's an amazing feature. How did you do? And the second thing about the notion of accuracy, right? And trying to keep accurate despite having a visual cortex. This actually, I stole it from Scott McCloud. So if you like comic books as much as I do, you probably know the guy. And this ability to actually identify and confuse an object with the representation of the object has fascinated like writers and painters all along. And I think this is one of the main tools and tricks used by people that write stories. And Scott noticed that you don't need a very strong accuracy or good representation to actually identify objects and extract information from them. Like, for instance, if I ask you, what do you see in the middle of my screen now?
Smiley.
A person? Sorry, I didn't get it. Can you speak louder?
Smiley.
a smiling person, right? Not on a smiling face. Not only you identify the face, but you were able to say that it's smiling, it's happy. But in reality, the only thing we have, is a circle, 2 dots, and a line, curved lines, right? Again, there's a trick because we tend to project what we know about the world on the image. And so we are not as stupid as a computer. We just don't see lines, but we see face. Well, this is not the subject, but the key, Scott actually noticed that, right? You don't need a great acuity, I don't say accuracy, but acuity in the representation to actually transmit an image. In the case of microscopy, that this is important, right? Because we are dealing with microscopy images, and microscopy, you're really at the edge of the small thing in the world. Like you really image the small stuff at the border of what you can image. It's very hard to go beyond microscopy. And so the acuity of the image we get, sometimes the images we get, they are not a good representation of the reality. They are strongly distorted. Despite this, it's okay. We can still extract like valuable and serious quantitative measures, but we have to keep this in mind. I will stop that with my digression with comic books and just sorry, ask you what is this? What do you see?
Fishes, maybe?
Sorry, I didn't get it.
Fish, maybe?
Fish? I notice our cells. That's fluorescence images of cells. That's the main difference with my students in biology, like in computer science. You see here, the stuff you see in purple, that's the nuclei of the cells. And you have some green dots inside the nuclei. Do you see them? Right? And in orange, you actually have the cytoplasm, which is stained by, I think I forgot, but it's a low resolution image, so everything looks confused, right? And so here you have a clumps of cells together. Actually, if I play the same trick that I did with Magritte pipe, this is not cells, but this is a spatial arrangement of not pigments, these types, but numbers. What's in the computer is this, right? We see cells or fishes, but actually not, we see numbers. And this is what the computer has in its mind or in its memory. And this is what Imaginize is dealing with. The core data with dealing this is numbers. So that's the data. So at one side, we have our visual cortex that tells us something, and at the other side, the computer sees that. And we need kind of to play with the data on the computer side to make sense of what they represent, visually, because everything is about images. And there's another thing I would like to point you is that, on this image, which is not a fantastic image, that's an 8-bit image over 3 channels, which is roughly this size in width and this size, in height and this size in height. So it takes, a certain amount of, space just to store it. Interestingly, if you do the analysis, I would say the segmentation of this image to actually extract, the cell periphery and which is what you see here in green and the nuclei in blue, then suddenly the amount of space you need to store this information is incredibly lower. And so there's the notion of information condensation, right? You start from something which is, let's say, a lot of data. It's very hard to memorize this amount of data. That's all the pixel value. And when you do image analysis, you turn to something like, which is, in this image, there are 35 nuclei and 33 cells. And so you have a huge amount of information condensation. And we say that you move from low-level information, the pixel data, the raw data, to higher level of information, like the amount of objects, their position, or their shape, descriptors, and everything. But the only thing is that this is useful for a scientific endeavor. That's the only thing that lets you draw a scientific conclusion, right? Okay, so we look at that. And if I have to reach a first initial conclusion, first another analogy for my use. Okay, I've completely forgot to tell you that. Assaf asked me to prepare a lecture to you, so I will absolutely not speak about what I do in my job, except maybe at the very end as a bad example, right? Bon, however, here's the counterexample. It's for my use. I was doing, let's say, biophysics on the cell cortex. So you see this gray ball here. You see it's a cell, it's floating. And because I was a nasty postdoc, I just made a hole here, you see. And so if you pierce a hole through a cell, you know, it compresses and you have a bulge that grows out of it. Or just for the information, you notice this bulge that grows is called a bleb. And if you're able to actually measure the speed of growth of this bleb and its size, you can learn a lot about, you know, the force generated by the cells. It's like, I don't have an example here. It's like squeezing, you know, a ball, and you have something that bulge. And so the image analysis task was just that, like, you know, measure the size of the bleb from the image. And so what we did was actually simply that. We take this raw image, we measure, we segment the cell with the bleb. And so this is what you see in red. That's the contour. It's a closed polygon, not even complex. And then on this polygon, because we know the cell is spherical and we know the bleb is spherical, we simply fit the union of two circles, right? And so if you think about it, the amount of information you condense from them is huge, right? You move from a raw image, which is not that big actually, to actually in the end only two, let's say 3 numbers. That's the only 3 numbers you need, the radius of the 2 circles and the separations. And you can get everything about, you know, the shape of the cell over time with just this. Well, however, like, you know, information condensation is not random. Like, you're not just going to say, okay, I have this amount of numbers, I'm just going to cherry pick and put it in my dissertation, everything's going to be okay. You need to have a goal, like what do you want? And then you need to have, I would say, a priori knowledge about the data, how to guide you to this information condensation. Such as, for example, I'm going to actually trash some pixel values out because they are interesting. So I'm going to do image processing or filtering. And then I'm going to lower the information about that. But the image is going to be easier to process. After that, I'm going to do segmentation. And our segmentation, that's the process of extracting object from an image. And then instead of having, you know, the image itself, I just have the contour, which is simply stored as a, you know, in that case, more than 300 couple of XY coordinates, the polygon in red that you see. And then finally, on this, I will fit 2 circles and then I have 3 numbers. But I could do that only because I knew what kind of noise I would find in the image, so that determined the filter I wanted to have. What were the features, by say texture, that would actually define what is the cell contour and exploit them to actually get the contour. And then finally, I knew in advance that the cells would resemble 2 circles because actually someone did the theory for me, right? So it's kind of important when you do that, that you have a priori knowledge in computer science, which is something you will hear me say again a lot in the following 1 hour and a half, is that if you want to do data science on, you know, microscopy images, you need to know data science, but you need to know biology and you need to know microscopy a little bit, which is, and that will be the conclusion of my talk, why it's the greatest job ever. Okay, so making sense of image, it's about that, right? So that's our task, and we're trying to actually quantify the image, which is a big word to say we have an image, trying to make sense of it by actually finding objects and actually assigning quantitative numbers to that. And for that, the key stuff is that you need a priori knowledge. And you will see every time you need to know in advance what is a cell, what was the staining, what was the microscope, and so on. Okay? Now, the second part, I'm going to use an example. If I understand correctly, last week you were trained in microscopy, correct? Please say yes.
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 Good. So here I wanted to measure something about the nuclei I have in this image. You know, that's the center of the cell. And so I stained my cell with DAPI, D-A-P-I. Do you know what is DAPI?
No.
No. It's a dye. Like, you know, the problem with when you're trying to actually visualize biology is that most, if not all, the animal cells are transparent. And so it's very hard to see anything. So sometimes we had dyes, such as fluorescent dyes, that will actually specifically label part of the cells. And DAPI, it's a chemical, like semi-dangerous, I would say, that will actually go to the nuclei on the cells, only on the nuclei. And so if you put DAPI on your cells, boom, suddenly only the nuclei start to shine. And so this is what you see here. I acquired the DAPI of my cells. And if I look here, if I zoom in this portion of the image, this is what I get. A bunch of numbers. And actually, you know, the image visualization know how to turn these numbers into an image, and we see that. Alors, my goal is going to extract the nuclei. How am I going to do that? Well, it's very simple. I simply, for instance, you know, that's an example, that's not always the rule. I simply say, okay, I'm going to set a threshold. If the pixel value is above a certain value, then I'm going to say that the pixel is inside the nuclei. If it's below, it's outside. Here I'm lucky because the feature that describes my biological object is very simple. Because the dye only goes to the nucleus, I simply have to measure where the pixels are bright, because this means where the dye is, so where the nuclei is, right? Sometimes it's not that simple, but in that case, and that case with fluorescence microscopy, is very simple. The pixel value directly encodes the presence of the structure of interest. And so actually thresholding is a simple way of classifying pixels which are either outside the structure of interest or inside the structure of interest. Right, and so with just threshold, you condense the information a lot. You move from a grayscale image to a black and white image. And after that, you won, right? You can label objects separately and say, you know, touching pixels belong to the same object. And then you get 4 nuclei there. You can have the boundaries and you can make measurements on a single object. That's segmentation. That's one topic in image analysis. I'm sure you know about it. And we're actually trying to extract objects from images. And once you have that, you can get quantitative information such as the size of the pixels, the object, the perimeter, the eccentricity, and the orientation, right? That's the ultimate information condensations we wanted to do. So it's impossible not to cite Lord Kelvin because he says, like, you know, quantitative information about a phenomenon of life, you know, physics or biology is the ultimate goal. You know, that's my conclusion. Image analysis, actually, more than anything, it's a way, it's the best way to actually extract quantitative information from images. And that should be our goal. We want to know when, how, where, quantitatively. Our answers are not only qualitative, but also quantitative. Well, here I just took the example of segmentation, but that's not the only way, right? There's stuff like colorization and something like this. And after that, in your case, I don't have to convince you, but sometimes, you know, the biology people you will meet, they will sometimes not be as good as you with computers. And so your work is about, you know, being good with computers. And why are the benefits of working with computers? What's the interest? In our case, we do science, right? So we are with scientific people, and our ultimate goal is the truth. And the naked truth, and not altered by any kind of objectivity or anything. So let's say, you know, let's play a little game. So this is a fake experiment, so in a fake article. We are trying to determine whether, you know, one protein, we call vimentin. Sorry, I'm trying to move the screen there. In red, is located at the same place that of another protein called actin in green that you see, right? They were acquired consecutively with 2 different dyes. The first actin in green, and then, you know, the second one in red for vinculin. And so you have 2 channel images, and when you overlay them, they look like this. And the question is, every time I have vinculin, do I have actin? And so the guy, let's say, you know, that the writer, the scientist writes this. It is obvious from these images that vinculin is co-localized, that's the term we use, with actin. And then another way to write the same thing would be the following. 93% of the vinculin signal is found to be co-localized with actin. A comparison with randomly scrambled images reveals that this number is significant. If you, dear students, would be to judge what one is better, what one would you find the most convincing?
The second one, the right one.
Obviously, the second one, right? Because well, myself, well, it's probably because I'm French, but I'm sure it's the same for everyone. Every time someone starts a sentence, right, it's obvious that I have these defense mechanisms that say, why is it trying to intimidate me or what? And so I tend to react with that, right? The main advantage is this, right? We do quantification because it's objective. It's the extraction of numbers that do not depend whether what you think is obvious or not, what you think is right or not, or what you need to be right. Right? So it's very important. Computers are objective. And so they allow you to distantiate the truth from what you need to be the truth. Next thing is this, that's a data set. All of these actually are single images. The good thing with computers is that I can write a script or a program that will actually do batch analysis. Right, so I will spend my time writing a script or everything that will analyze everything in batch. In the meantime, I can enjoy coffee. The harsh reality is that sometimes biologists, they do this one by one. Like different skills, different cultures and everything, right? And so one of the other benefits with computers is that they're very good at automating repetitive tasks. And so I think that's why on top of actually being good with everything, you need to be good with computers. You need to use them, because they're objective, and that's super important. Like the pressure on people is so huge to have positive results that at some point you need someone very stupid, such as a computer, to actually tell the truth as it is. And then, it's also a good tool, a good friend, because it will automate tasks for you, right? As we speak, I have the other computers running an analysis, and I don't even have to think about it. Okay. And the last stuff I wanted to say in this part is again, taken from my youth. Here it's a little bit complicated. You see, it's a weird experiment I did with the lab of Carl Philipp Eisenberg and Jean-Léon Maître when we were in Dresden. So you see here, you have two pipettes. And so at some point, I was good with actually building microscopes and microscope equipment. And in these two pipettes, I sucked, two cells, progenitor cells of a zebrafish embryo. And they stick together and they pull on each other, the cells. And it's very important, to see how they generate contact protein that will help them actually sticking to each other. And it's super important, for how you form tissue, right? Because of course, like cells have to move, but they also have to stick together. Look at this movie again. Right, so I was putting the two cells in contact and then watching over time, how the area of contact is growing, going from this to this. And the speed at which this area grows actually tells you, how fast contact molecules are recruited. And so that was our task, actually, measuring, the area of contact over time. So that was our strategy. So we were going to speak about strategy for analysis. These cells, they image in bright field. Do you know the difference between bright field and fluorescence microscopy? You have the right to say yes or no this time.
Yes.
Who doesn't? Very briefly, fluorescence, use the dye, like the DAPI I showed you. And so you only see the area of the cells which is stained by the dye. And so this is why you have something which is black everywhere except where the cells are. In bright field, you have your cells somewhere, and then you shine light through it and you collect it above. And so everything is gray or white. And so when you actually go through the cells, you see you have these dark stripes here. And so this is why you get this kind of black stripes around at the periphery of the cell with this aura around it, right? And so we have a very dark perimeter. And our idea was actually just to actually take two circles and adjust it so that, the perimeter would actually fit the the lower thumb of the pixel value. And so with that, we would have, a rough idea of where the cells were and the size. And from that, we could actually measure the area of contact. And so that's the curves you see here. We were tracking the contact length, the size over time. And I think, in blue, you have the automated measurements, and in red, the manual measurements, the ground truth event. It's not bad, huh? Right. Do you think it's really how it happens? It didn't happen like that at all. Not by this method, like say, oh, it turns out that we received images that are in bright field, and so we derived the technology to, a pipeline to exploit them. It worked in reverse. Jean-Léon told me, I need to measure the contact area over time. And then we started to brainstorm and say, hey, Ton, if you want to do that, you need to know the cell position and their size. Okay, so to do that, we could fit a circle around each cell. In that case, if you want to fit a circle around each cell, you need to have an imaging mode that outlines the cell contours. So we could go to either fluorescence microscopy and have something that stains the cell membrane, or to bright field. We picked bright field because we were lazy, because fluorescence, you have to add a dye. And so we just did that, right? And so every time, remember that bioimage analysis, it's one step. The part with computers is one step. It's something big. And you sometimes have to go back to the guy that prepared the samples or to the guy that imaged them on the microscope and say, I would need to do that, right? It's really an integrated approach. And again,
For me, it took, I think, 2 years to understand. So my background is, of course, also computer scientist. And I think to understand that you actually can control the data generation and ask your collaborators to do something a little bit differently to make your job easier or to ask a new question, it took me about 2 years to realize that. So I was used to, going to data sets, and what you get is what you have, and finding patterns within them, and being able to make this distinction, it's not trivial at all, for me at least.
But would you agree? Is it better now?
Yeah, much better. This, and I think this is one of the big advantages of the field, because as a data scientist, usually you go and you cannot control things, right? You go to Twitter and you have a network, and you just have a network. But when you have your cells, you can actually change something within your cells, and you can control the change in the network, and then understand that, right?
Well, that was the whole point of the lecture. I think I could stop now. Also, this is why this job is so interesting, right? Because you get actually to talk to microscopist people, and you get a chance to actually play with image quality, right? Because sometimes you get images of really poor quality, and the ability to say, right, let's use another strategy for imaging because it's not going to work is super interesting. Although then, personally, you get a chance to see a lot of biology, right? Because, this is development, of cells from tissues. But in my job, I also get a chance to work with AIDS, and I'm going to come that on that, with COVID or with neuroscience. And you are forced to actually get cultured on that. And it's why it's super interesting, right? At some point in your career, like I think we should speak careers, right? It will be important that you pick a job where you won't be bored, particularly in your 40s, right? Bon. But that was the point of this part, like making sense of image, and you have to think a little bit about strategy. The numbers you want actually determine the image techniques you will use. It's not the opposite. You don't receive the image and you try to see what numbers can I get from that. It's the opposite. Set a goal and tell the guys, I need this kind of image with this kind of quality. So yeah, we have to be a people person a little bit. Okay. Now, instead of making a break, I would like to make a small interlude. Right? Image analysis, it's a subfield of applied mathematics. So you have to be good in mathematics, which I'm not, let's be frank. Is this recorded, Assaf? I hope it's not recorded. I'm going to say stuff that are embarrassing.
It is recorded, but it's fine. You can check my recordings from last year, and I tracked, the whole, yeah.
I will make beeps to this part, right? But however, despite the need for applied mathematics, we still have this visual cortex we could exploit in some kind of intuitive things. As Scott McCloud says, we have this tendency to immediately identify objects, and you can use this to actually derive an approach for image analysis, asking you the simple question: why do you see something? Not taking your vision for granted, but as a beautiful, perfectly evolved tool. Here you see there's a cell, and you see here there's a small blob. Do you see it? The question is, why do you see it? What, okay, translated into vernacular world, what are the features that allows your brain to say, here's the cell, and here this is not a cell? That's the background.
Different color.
No, there's no color, it's a grayscale image.
Boundary.
How do you find the boundaries? That's exactly the question. What did, what allowed you to say that, you would say that, this is the boundary. Do you agree with me?
The boundary is like a bit darker from
Change in the change of the gray level, the gradients.
What?
Change of the gray level.
Is this Anatoly?
Yes.
Right. So you're on to something, right? It's the changes that matters. It's not exactly the pixel values, right? Because they're gray everywhere. Here it's gray, here it's gray as well. It's just that when you're inside the cell, it varies from one pixel to the neighbor pixels a lot. While when you're outside the cells, it's flat. Now, there's a reason for that, right? When the light goes through the cells, there's plenty of things that are in the cells, like vesicles, polymers, the nuclei, and so on. And they send the light in all directions. So sometimes you collect a little bit more light because they act a small length, sometimes less. And so when you are in the cells, there's this texture or the variation. But when you're outside, if your sample is clean, obviously nothing will change the light until you have gray. So as soon as you know that, the local variation of grayscale set what's inside or outside the cells, you can use whatever tools actually tells you, okay, here I have high gradient value, here low value gradient, and then you get this. Good? So you use your intuition to do that. Next question. That's okay. I took very limited number of examples. That's the same, let's say, data set. That's a cell, and I'm passing a hole in the cell here. You see here the bulge, the bleb grows. But if you see what happens at the back, the cell retracts. You see, and so there's a movement from this part here. You see the movement? Zoop. And my question to you is, okay, I see a movement. I hope you see a movement too. Why do you see a movement? I know that this technique is very dear to Assaf, so I forbid him to give the answer.
Because change of the gradients.
Ouais, okay. The first part is correct. And can you use maybe a better word for the second part?
Yeah, places where a lot of changes, you have now gray. It's just a regular gray.
I'm demanding, but one thing you could say is that you have a certain pattern, like the texture has a certain pattern, and in the frame next to can I pause this? If I find how to pause it? No, I cannot pause it, right?
You need to go out of the presentation, I think, too.
Well, never mind. Actually, I can use that. You see a certain pattern, and in the next frame, the same pattern, or close to it, happens again, but in different place, like this. Right? For instance, you have a well-defined pattern here, but later in time, roughly the same stuff in a box is found here. And so what your brain does is that it extrapolates, like in a comic, when you have still frames, your brain extrapolates and say, plot, this green row is what happens in your brain, and creates a continuity like that. And so this is how we detect movement, actually. We split, our vision into small boxes. And every time there's a pattern inside these boxes that moves, our brain say, hey, it's the same stuff, but that moved. Like if I do this, you will see it moves, but basically just the pattern of my head is in here, 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 and that. As soon as you know that, there is an image analysis techniques that actually exploit just that. That then you actually takes a pattern, split the image into boxes, and try to find where it is in the next image. In that case, this is called optical flow or particle image velocimetry. It's another implementation of the same goal, roughly like this, right? And then you can have this velocity vector. They're much more accurate than just seeing things move, because you get an information of exactly where, measurements of the angle, and the speed. And by the way, if you're into streaming or watching videos, this is how the MPEG-1 compression standard works. So, okay, so every time you try to use your intuition. In bioimage analysis, we work with images, and sometimes, let's say, our visual cortex can help us getting ideas. It's not always true. There are some part of image processing that comes from the darkest corner of mathematics, and are completely counterintuitive. Never mind. Okay, let's move to the second part, which are the typical challenges you will find in bioimage analysis. I'll just pick 4. That's the one I have.
Maybe it's a good time to have a 10 minutes break.
Or answer questions. Yeah, okay.
So maybe we go to a 10 minutes break, and then guys think about, if you have questions about the previous part, we can ask them, okay?
Okay.
Okay, thank you. Disappear.
Thank you, Anatoly, for answering the questions and participating. Super useful. Helpful. Let's go again.
Yeah, maybe we wait another minute.
Okay, okay. I'm going to make it, Assaf. Huh? You don't. So thank you I say thank you very much for being cautious with me, but I'm much better now. Just short of breath, but that's all.
Okay. I mean, yeah, I mean, yeah.
Let's go, people.
Okay, let's continue.
Okay. A few of the things you have to keep in mind when you're actually collaborating with someone, a biologist doing bioimage analysis. I noted 4 challenges, and how you harness them basically is going to help you have a good career. And so I just put 4 of them, which I have in mind almost all the time. And the first one is actually remaining objective and scientific relevance. Like, remember in the first part we outlined that we are scientists, we are obsessed with the truth and the naked truth, absolutely. And we do bioimage analysis because it's a quantitative, it gives you a number. And they are perfectly objective. The question is that you have to be cautious. If you want to have perfect objectivity, you have to be cautious because your analysis pipeline can be flawed a little bit. Here I just took a very dummy, a very simple example. Remember at some point I say, okay, I have an image of my nuclei, my 4 nuclei, they resemble like patatoids or blobs like this, bright blobs. And if I want to segment it, I simply have to say, every pixel with a value lower than 48 will be considered background or part of the nucleus otherwise. The question is, why did I pick 48, right? It's somewhat arbitrary, and it's something that I picked because it resembles what I think is a nuclei. The danger is that on this image I pick 48, maybe on another image I will pick something else, and the value I will pick, it will change a little bit, not much, but the size of the nuclei, the shape a little bit, and so on, right? And so if I decide what's the value for one data set and what's the value for another data set, I have a human bias and the truth is flawed. And so keep this in mind, right? You want to remain objective. So every time either you use the same value everywhere because you have comparative data, or you actually read a little bit. And you will determine that 48 you can say, I will not decide that, but there's actually algorithms that select automatically a threshold value. And they have hypotheses about the image, and if your image respects it, it's going to be valid, right? It's about hundreds of them. So keep this in mind. Next challenge is, like pathological results. We say computers are good because they're stupid, and that stupidity makes them objective, but it makes them very stupid. And here I have an issue, with object 2, 3, and 4. It's completely stupid results. Can you tell me why? What's the problem with this analysis? You don't see?
They are not complete.
Well, absolutely. You see, for instance, if you look at the area, all these 3 objects here, they have a small, very small area. It's simply because, the image cropped them. But the computer doesn't care. It just does what you told it to do. Right? And in the end, when you collect, imagine hundreds of thousands of these, metrics, there will be flow too. There will be a bias this time because the computer is stupid. So how would you, let's say, tell the computer not to include these results? You're a bioimage analyst, you're making the analysis pipeline, and you want to tell the computer to avoid that. How would you do that in practice?
You can take an average size of a known, let's say, to cell, and you can ask whether the other objects are in this range.
They're outside of the main distribution, right?
Right.
It's not great, because then you miss the opportunity to discover that there might be a subpopulation of cells that are small. The simplest way is simply to say, for instance, if you have an object that touches the border of the image, reject it. Right? And so you protect about that. And again, so keep this in mind when you're actually deriving an analysis pipeline. Always check what the computer gives you. Maybe not on all the data, but a little bit, so that you avoid these kind of problems, and add rules to avoid bias in measurements. Now, the third one, and we're going to talk a little bit at the end of this lecture, is the amount of data, seeing image as a data and data management as an issue, as a problem. So this is a very old screenshot from when I was a microscopist, 10 years ago. And so all of what you see here, each folder is actually a data set. And so each data set has quite some size, and all of them were acquired very quickly. Now, the microscope we have today, they're really able to generate a huge amount of data, really a lot. Up to the point when in the facility we faced an issue where the network was not fast enough so that the data could be transferred out of the microscope to the central storage. And that was, really blocking the microscope, something as stupid as that, for a multi-million machine. Right, so the data management is going to be an issue. And there's plenty of questions that are not related to actually image analysis, such as what's the best way to transfer it, to store it, to archive it, because you don't have the right to just trash scientific data. You must keep it for years. Sometimes, you can have huge image, really huge, particularly in microscopy. Or are you simply going to open it to visualize it and not even mentioning processing it? It's going to be crazy. After that, remember, biology, it's mainly nowadays a collaborative effort. So you will work with many people that probably don't have the same time scale as you. For instance, you can be the leader of a group, and then you can have a master student, and then the master student leaves, and then the data is lost because it's gone. He labeled the field he labeled the files 1, 2, 3, 4, 5, but you forgot what was 1, 2, 3, 4, 5, and so on. It's a mess. It's crazy. You see here, I put a skull, and that's really data management is horrible, really horrible. And so my humble opinion is that, the future, in biology, belongs to a good way of organizing data, and probably, scientific image databases. It's a very slow start, but I think it's important. But more importantly for you, if you want to be successful, regardless of where you choose to work, what domain, be organized. Number one features, number one skill that will make you successful beyond luck, talent, or how many hours you sleep, it's be organized. I am not organized, and I tell you, this is true. Right, and I just preparing this course, I said disorganization is the best way to kill a project. I have countless examples in my everyday life. Right. Well, and then I want to insist on that. The number of data, but also the size of data, it's getting worse every day because you're studying at a time where the progress in microscopy is exponential. Like, these two guys are microscopes. They don't resemble the idea of, classical microscope we have, with the two oculars and the moving stage and so on. They are highly performing, very speedy machines that can acquire a huge amount of data. This is, from my work with the lab of Philippe Sansonetti, when we are trying to actually image the bacteria, the Shigellosis disease. It's one form of, in French, we call that la tourista. You probably don't know that in Israel. It's that, when we have French people that goes to tropical countries for holidays, they drink the water, and then they get some kind of food poisoning that destroy their guts. And so Shigella is one of those diseases. It's a bacteria that goes in your gut. And so what we wanted to do is actually image like the 15 centimeter of the gut of a guinea pig and every single bacteria. And of course, the data is huge, but it gets worse, Okay, that's just an example, that's one excerpt of the image. And if you zoom very carefully, you see these 4 dots, green dots, that's a single bacteria. And so this is a 3D image of that. This is the 10 centimeter, like not 15, of guts there. And we could count every single bacteria there. Now, of course, the data is huge. For a single image, the data is really big in size. But this is nothing. Nothing compared to what I did with the lab of Pavel Tomanchak. And so here, that's developmental biology. And here you're watching, actually, this is an egg of a shrimp. Apparently, Hawaiian, that's a small shrimp. And these guys, Karsten and Anastasios Pavlopoulos, they actually imaged the shrimp every 7 minutes for a whole week. And all the blobs you see here are cells. Look at the results. We move from something that is spherical. Here you see the progenitor of the head, like this, right? And here you see digits emerging. You see them? I work on this, but I did not the microscopy. I find this amazing. Look at this now, like all the digits you have, antennas, like legs and so on. And you see every single cell of this organism, you see it. And look at the time scale, like, we're getting close to a week, like several days and so on. This is one single movie, and the raw data set is 7 terabytes. I have a decent computer at home, and even with that, I would not be able to store it. And so they acquired this data in 1 week, but we have been working on it for years, just trying to build the tool that could open it. Right? And that's one of the challenges we will face, the size of the data. I'll speak about it later. And it's fantastic, right? Well, let's just repeat that. We can go in detail. Why do we know we have huge data, but it's not super great? And then the final challenge I listed here in this short list is the need to go beyond. Image analysis, it's not the end. You will not be flawed by this. The stuff is that image analysis, it takes an image and gives you numbers. And so, the information is really condensed. The problem is that if for a data set you have thousands of millions of images, you get thousands and millions of numbers. And you still need to condense them more to have a scientific answer. And so for this, you need to master statistics, right? Interpret all these numbers to decide whether yes or no, I have a phenomenon or not, where yes or no, the difference between a mutant and wild type is significant. And so you need to be good or at least known statistics. So you see, at some point, there's biology, microscopy, you're in the middle with image analysis, and then on the other end, there's statistics. So you can choose to collaborate with people that have these skills, and it's not or, and you need to know a little bit of everything. Are you guys trained in statistics? Do you find it fun? Like experience may vary, right? Sometimes. Right, I find it that I repeat, that's why this job is so great, because you get a chance to have go really wide instead of being very tight, right? Voilà, that's just the 4 typical challenges we have. I wanted to underline it in this short introduction, right? Bon, but I'm glad because I'm well on time. Do you have questions on this part before we move on? Good. Okay. Assaf asked me to speak about ethics a little bit. And again, I have the scientist approach, and I told you about the absolute relationship, an extreme relationship to the truth, which leads us to speak about scientific misconduct. And so I hope that in your career, you will never, ever be confronted to that. But just in case of, we thought we would speak a little bit. What is scientific fraud or scientific misconduct? Or let's say simply cheating or treachery in bioimage analysis. I would like to outline the notion of personal impact, different level of image manipulations. I will not tell you don't do fraud because it's obvious, but I will try to teach you how to protect yourself from fraud, and then have a little bit of, let's say, fun or a small game using the work of Elizabeth Bick, which is really worth noticing nowadays. Alors, this is going to be tricky. That's one of the articles I published. I didn't put it here just to show off, but just to show you something. That's an article we published. It's a collaboration in 2019. And you see here, I'm listed on the author, and Jean-Yves Tinevez, I can tell you, this is my real name. I'm really called like this. The house I live in is under this name. I have 2 sons, and they have this name. And that's the name of my father, my cousins, and everything. You see why it's important? Like maybe there's another case. Do you know a French singer called Johnny Hallyday? Who knows this guy? Probably not very well known in Israel, right? So he's a famous French singer. Sorry, my voice starts to go. He's really famous. He's a singer, and he sold millions of discs and songs. But it's not its real name. Its real name is Jean-Philippe Smet. And so for his work, he has, I don't know how to call that, a makeup name called Johnny Hallyday. And so it's a great advantage because if he ever, he's dead now, never mind, but if he ever screwed up his career, that will be the career under the name of Johnny Hallyday that will be screwed up. But the person called Jean-Philippe Smet will be fine. In science, we don't do that. We use our real name. And that the problem is that if you ever make a big mistake, a fraud, a scientific misconduct, this paper, for instance, this paper does not contain scientific fraud. It's my best work, I swear, right? It's just an example. It will be retracted with a notice like this: inadequate and intolerable scientific fraud. The first author is a fraud and a loser. Well, they generally don't put it under this term, but this is what everyone will think about when they will go to this paper. It's going to be public. And so this is still my real name. So that's the name I will use, because then my career in science is over, obviously. But that's the name I will use to apply to other jobs. And so the guy says, Jean-Yves Tinevez, are you the guy that actually cheated on the Nature paper? Oh, I know that, right? You see? So when you actually produce scientific misconduct, it has a very deep personal impact, because it associates everything about you, your whole professional life and even personal life, to this. That's the danger we take in science. We use our real names. It goes beyond that. I have children. And so let's say if I make a really big fraud, something that will last for years, when my kids will apply for jobs, says, are you related to the big fraud in nature? You see what I mean? So scientific fraud, very bad idea, right? It's generally a better idea to say, if something doesn't work, if really you feel that nothing works and that's the end, that's fine. Just get another job and you're going to be fine. But scientific fraud is never an option. And so, unfortunately for us, it occurred more and more in biology. So there are not a lot, actually, we have to right to be optimistic, but there are some scientific fraud in biology. And so, including in imaging. And some people have tried to actually come with recommendations because the biologists, they say, okay, I manipulated this image, I altered the truth, but I was not an expert in microscopy. My field is biology. And imaging is sometimes not seen as a biological field. And so people have started to produce guidelines. First one was this paper in 2007 by Cromie, sorry. It's 12 guidelines to avoid honest mistakes. And so you will see that they need an update, right? But so far, so good. Scientific digital image are data. That's super important, right? That's scientific data. So you don't touch it, you don't play with it, you don't compress it, you don't alter it, you don't trash it. It's precious data. Manipulation of digital image should always be done on a copy, right? So you don't alter the raw data, which is as close as the truth. And so you see that the raw data in this philosophy becomes quite precious. Simple adjustment to the entire image are usually acceptable, right? So when you display an image, display is not raw data, so you have the right to change something and so on. Cropping an image, like taking a small except, it's usually acceptable. And then I started to dislike this a lot. The reason is that that's my personal opinion, right? So not everyone agrees, and it's okay not to agree with me. The recommendation of this is, in my humble opinion, not okay. The reason that is, is like this: manipulations that are specific to one area are questionable or not recommended, and so on, and so on. What I didn't like in this, still don't like with this recommendation, is that they're very vague, and sometimes they move from something which is oddly specific to something which is super vague. Like simple adjustments, what are simple adjustments, are usually acceptable. Acceptable means it's okay, but not completely okay. Also, you can be scolded, but you will go through publications. I don't know. Cropping an image is usually acceptable, which is, okay, are there times when it's usually not usually acceptable? I don't know. And I think that, if we actually push this too much, we simply discourage people, from working with images. It's so vague that when you read them, you say, okay, I have no idea what is scientific fraud, so I'm going to read this paper to have an idea. And at the end, you say, I still have no idea what is a scientific fraud. I still have no idea what to do. So I don't like it. What I will try to show you is that you can work around it by knowing what is data and what is scientific data and documenting what you do. Right. And so, for instance, not every mistake is an honest mistake. Now, there's another paper, this time a bit older, by Rosner and Yamada, which is the temptation of image manipulation. Well, I will not comment about why is it a temptation. We can discuss why people do that. Do you know this kind of grants called gel blots? Have you met them? Never, Assaf?
I did, but the class didn't.
Okay, right. It's a classical, very, very typical technique in biology when they actually put proteins into a lane where they would go down. I'm terrible at explaining, forgive me. They will, let's say, position at a certain place that gives information about what they are and their size, right? And so they show up as these dark bands on a lane, right? So this horizontal stuff would be called, a lane, and so this is a band. And so the image, for instance, that's an example. the image that was published is this one, but people say it's suspicious, and then they ask what was the real data, and the real image was this one. And so the author of this image simply says, I have a band here. In my view, it shouldn't be there. It's a mistake, or it doesn't fit with what I want to say. And so the guy took Photoshop and erased it. Is that okay? Obviously, it's not okay, right? It's a complete change of the truth. It's saying you're hiding the truth, right? Even if this is annoying, you don't have the right to hide it. And sometimes the opposite happens. You say, the guy says, you see here, I have a protein that's missing. I don't know what was wrong, but there should be a protein here and it's missing. So what the guy did is simply, took Photoshop and copy-pasted this line here, right? So these two, lane 2 and lane 3 are completely different experiments, but the guy actually copy-paste. It's not okay at all. Again, this is fraud. That kind of things means the end of your career, like the stuff I showed you in the introduction. These are the consequences. Something which is more subtle, like this is electron microscopy, so it's gray everywhere, but they have techniques where they can actually label some structure with gold particles. And even in these tiny things, tiny scales, these gold particles will appear as very black, very dark spots. And the authors wanted to label like some proteins that were stuck on the membrane of this object. You see here, you have two vesicles. It's very hard to see, right? They're very faint. And then you have spots there. And in the spots is the result of the label. Do you see them? Please say yes. And so the guy said, yes. If I show that, people will comment, right? They will say, it's too dim, and then I have this big black spot here. It shouldn't be there. It shouldn't be in this part of the cell. It should be there. So what the guy did is that it simply erased this, right? It disappeared from the manipulate image, and then it simply locally changed the contrast. It's enhanced it right now. So everything which is gray here, it's black here. And so this is what you do on your family pictures, right? Sometimes you want to remove uncle you don't want to see on your family picture, or you want to actually say, oh, I'm too dark. I'm going to change the lighting so that I shine on this picture. It's okay. It's totally okay on your family pictures. When you're trying to uncover the truth, like when you're doing science, it's absolutely not okay. End of career. Let's go on a little bit. Voilà. Change the contrast of brightness, made only of part of an image. Not okay. And then after that, we have stuff that are weird. So this image looks fine, but actually, if you edit a little bit the contrast, you will see this. You see here, I changed the contrast a lot to actually show what was the background. So I completely saturated the display. And you see this black area? So what the guy did actually, he erased this part of the image and actually copy-pasted cells from another image there. Then it's the opposite, right? You have someone that's missing on a picture. Let's say you have a picture of yourself and you want to put David Bowie on the picture to show that you met David Bowie, right? And so you copy-paste David Bowie from this picture to use, and that's obviously not okay. Now, this case is particular because the person that did that actually copy-pasted cells from an image of the same data set. And so in theory, it didn't change the truth, right? Because it was showing cells from the same image. However, no, this is not okay. This is altering a raw image and presenting it as raw. That's not okay. It's like lying, right? So don't do that either. And then there's plenty of examples, like this is again bands, but every time you change the color scale, the lookup table, or the contrast and brightness, it's very easy generally to see manipulations. So you see, that's a time lapse, 3 minutes, 5 minutes, 10 minutes, 30 minutes, 60 minutes. These are embryos, but you see that at 3 minutes and at 30 minutes, that's the same image, copy paste. The same from 0 to 30, copy paste. Probably, maybe the guy lost the image or forgot tune the microscope on at this time and fabricate it something. It's not okay. So there are simple tools to detect image manipulation, and generally they are not very strong. Final story with that. It's not in biology, but it's a famous example. In science, material science, there's a lot of efforts that are on synthesizing micro or nano materials that have well-defined geometrical shape. And for instance, this guy called nano chopsticks. So you see, they're very small. That's like carbon structure, and they have a very good, very fixed angles between the two structures. I don't know why, but it's super important for these guys. And so they're trying to synthesize chopsticks, that's two rods that have a fixed angles between them. And if it do that, it's very big for them. And so you see here, how does it look? It looks good. I have managed to synthesize something which is close to 90 degrees. But now if you zoom to the image, do you see? You even see the rectangles that were copy-pasted. Actually, it's not a true scientific image, it's a synthetic image. The guy actually took the image of a rod and then copy-pasted it and copy-pasted another. And so the paper, there was an investigation about that. And now if you go to the paper, you see this. Chopstick nanorods tuning the angle between pairs, and now you have this withdrawn. This paper was withdrawn. So you see the stuff I showed you with the first author is a loser and a fraud and so on. They don't put it like this, but everybody knows. And again, this is generally very sad stories because these guys, that's the real name. And it's going to be very hard for them to have like funding and so on. It's very hard to recuperate from that. So really stay away from that. Every time you see some suspicions, of inadequate manipulation or see something odd, something that tickles your ethical sense, every time you see, okay, we're actually playing with the truth, go and talk to your boss. Never go there. We okay with that? Do you have questions about ethics? Like the question is how to avoid doing it. I'm sure nobody wants to do it. My practical recommendation, my practical suggestions, it would be the following, right? How to avoid making inadequate manipulation is that every time you do something in bioimage analysis, you write down everything. And when you know that when you publish an article about a finding, you have to write the material and methods. You have to describe everything you did for the experiment. And so what we do with colleagues is actually, we try to do that, right? We describe very well how to produce the analysis results. Like for instance, compare something like, images in panels A, B, and C were acquired with the same settings and have been linearly adjusted for contrast with equal mean and max. So we say exactly what you need to actually make this plot. And then compare with someone that would say, a band in lane 2 was manually removed. The third lane was copy-pasted from another study and used as a control. This with the law, this is not okay. But if you write it down, it will allow someone to say, hey, that's not okay, you cannot do that. And so you will detect the problem early. And so we go more and more in these directions. And so now we tend to even publish not only the findings, but also the methods. And you feel that. You will sometimes see that in scientific paper. The scientist actually says, the raw data is at this location, the Jupyter notebooks that does the analysis is at this location, and if you run it, you regenerate the figure. And so that brings you to actually the notion of scientific reproducibility. Assaf and myself, we hear that all the time, the reproducibility. Every time we have publications, we are asked about it. But did you ever met this notion, reproducibility? Have you heard about it before that? You're probably too young, right? Right, so that's the key message. If you're unsure about scientific fraud, simply write down everything. Be 100% honest. I don't dare as a computer. Okay, last paragraph in this part, I would like to outline the work of Elizabeth Bick. Assaf, you know her. right? She's becoming famous, and also infamous for certain, because she's on her personal mission is actually against scientific fraud, and in particular in imaging. And she made plenty of findings about, there's even companies that specialize in scientific fraud. And she discovered that. And she animated a funny Twitter account where she said, here's my findings about manipulation. Help me discover that. And so I asked her if I could use her work for this lecture, and she said, okay. And so in the next five minutes, what I suggest is that we do that. I'm going to show you some pictures, without context. You don't need to know biology about it. And your work is going to find out what's the problem. 1 point for the first guy for the first person, sorry. It's super easy to find it.
There's not supposed to be 2692 in Bioimage.
Well, the problem is with this band, but actually, if you see, this is the exact same gel, except that on this one, this guy was deleted, or the opposite, right? We don't know. Maybe this guy was added, right? But this is supposed to be 2 experiments. And so you have 2 completely different experiments that give you absolutely pixel-wise the same image. Impossible. It's a fraud. Okay, good. 1 point for Samuel, that was you? No? Bon. Alors, this one's difficult. Actually, that's a famous example. So what you see are colony assays. So we are trying to grow yeast, actually, or bacteria, or whatever. And every time they successfully grow, they make this small black dot, right? And so it's a black or white experiment. Either you get something like this, either you get nothing, and there's no black dot. And so this picture was actually used 3 times in 3 different papers by 3 different groups that don't even talk to each other in 3 completely different subjects. I think this is infection diseases, this is genetic disease, and I think this is cancer. And the authors are not even the same, but you see this guy is the same that this guy. All the red boxes are the same. And see, it's kind of a combination. And so this is how Elizabeth started to suspect that there are people you can pay and they will write a fake article for you.
Oh, wow.
But of course, they have a limited amount of images, and so they will simply reuse, combine. And sometimes they stretch it. So you see, this is circular, and here it's not circular, it's compressed, right? Next game. It's a cell death assay. So I have 3 different experiments. Control, I do nothing. NC, I add something which is supposed to be any effect. And in the 3rd case, I add something which is supposed to kill the cell. Can you see what's wrong?
Control and NC are the same.
Well, absolutely. 1 point for you too, right? So how is it possible, right? Because this is supposed to be 2 different experiments. Fraud. Oh, this one is very hard to see, but that's because of the quality of my photograph.
Control liver and liver 40 are the same.
Assaf, can I give the control to someone? Remote control.
Yeah, I think you can. I'm not sure how.
I know. Who wants to try? Samuel, was that you?
Yeah, yeah. So
okay. Shit. Sorry. Zut. I cannot give you control without restarting.
it's okay.
Crap. I'll think about it next time. Well, but basically, you probably see, right, this image is the same here. Which is funny because our visual cortex is actually very good at finding similarities. And so this is why I put it on easy. Right? And so for 2 different experiments, the person actually used the same image, but shifted a little bit so that we wouldn't find it that quick. Now, this one is difficult I think I found it. Did you find it? No?
Is it the same image inverted?
Not inverted, actually stretched.
Oh, yeah.
So this image actually is compressed like this, but it's that one. So that you deform. Okay, so this cannot be an honest mistake, right? It's not a mistake by I accidentally renamed the file or something like this. The guy actually manipulated the image so that we wouldn't find. Okay, this one's difficult too. Look at C and D, which is supposed to be two different persons.
Do you see only the bottom is removed?
Ouais, it's rotated and the bottom is removed. Now again, it's impossible for this to be an honest mistake, right? The picture was composed. And then that one is I know this one is what I call la fiesta.
Oh, I can see this nuclei at the lower left side, which is practically the same. Ah, it's actually duplicated multiple times with the two dots. Yeah.
Well,
Yeah, even in B.
Oh, wow.
It's a synthetic image. You know, the guy actually took one nuclei and then pop, 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 stick it everywhere. Okay, so almost at the end. So you see, this is a fluorescence image, the stuff we have the most case, you know, black background and luminous stuff. This one is tricky, actually. Nope. It's like this. You also have like a repetition of the patterns that you see here. You know, several times the same nuclei, and you have an image which is in common, but that's been stretched again. But again, you know, this guy and this guy are two different persons. Okay, and just to conclude, my favorite, what I found, no, sorry. Ready? Okay, so this one is difficult because the image quality is not great as well, but this is again an image that was synthesized actually. And the effort the person put into synthesizing this image is huge. It's really like hard to, you know, actually copy this part here and here without the ability to detect that. The guys really put energy on that, but not on science, obviously. And then my favorite. These are, you know, two different, that's two photographs of the same person, but, you know, combien? 6 months after treatment, right? And this is about a treatment that's able to remove skin lesion.
The mole is missing?
It's worse than that. This is exactly the same picture, but the moles have been edited out. Right, and now I got my hands on the image, and you know, you can align them, and they perfectly match the pixel. It's impossible that's, you know, the same person, but 6 months after. And so there was investigations about, you know, scientific misconduct, and for the defense, the author says the photograph in our article is the same patient at 6 months post-treatment, but he simply wore the same shirt 6 months after that. Right. The issue with that is that all of this, you actually played with the truth, right? And so in your paper, you made a statement, but because because these guys played with the truth, it's impossible now to decide whether what they say is true or false. because they played with the evidence, they play with the raw data. And so that's why actually we did this exercise. You must protect yourself from the fraud of others. Because if you ever actually base your research on the data these guys generated, you might lose a huge amount of time. You might lose years. You say, it's impossible. I don't understand. I'm unable to reproduce the results. I must make a mistake. And maybe, no, you're not making a mistake. Maybe actually the guy committed fraud. And this is why people are so angry against scientific fraud. Because in science, typically, we say we stand on the shoulders of giants. We actually, use the discoveries of others to go on. But if the others have cheated and manipulated or made scientific fraud, you have a problem. And so, when it comes to ethics, it's also defend yourself and try to actually always act as a suspicion person, always try to expertise the image, look at it and say, is this okay? Is this objective? Is there manipulation or something like that? Because you will have a problem with that. You okay? It's not a funny subject. I think it's always sad because it's thing about shattered career and problematic manipulations. But someone must talk to you about it. Are we good?
Are these examples gone through peer reviews?
Yes. And they escaped it. That's the talent of Elizabeth Bick. The problem is that, you remember what I told you, she found that there were some companies that can produce fake articles for you. She called that a paper mills. It's a mill you go and they give you papers. But these guys are very good also at faking reviews. Right, and it's horrible because there's now a business in fraud. Apparently a profitable one too. Right, so my main conclusion is that, right, I am sure you will not commit fraud because you're aware of the personal impact it has. I'm just wary of, I want to make sure that you will protect yourself from the fraud of others. And I hope it will disappear. Bon, but this grim part must not let us forget that, otherwise a part is very noisy minority. It's a tiny minority. And otherwise the people you will find in science, they're all animated by this extreme relationship to the truth. And you will have no problem with that. Good? Right, okay. I'm almost done. The last two parts are actually very small. We'll do that quickly?
Mm-hmm.
Okay, Assaf asked me to actually prepare a small presentation of the different uses and careers in bioimaginize. So typically, so that you can identify the kind of person you will collaborate with in your collaborations with biology. I just want to say it's a little bit like for data science, if you think about it. So data science, it can be seen as a research endeavor, right? So there are professors and university chairs in data science. And so in that case, the people, the researchers that work here, they do research on data science. Data science is the focus on their research. And so their goal is actually they pick a fundamental scientific question that advances data science. I don't know because I don't do that, but it can be creating a new algorithms, or actually discovering a scientific understanding, of concepts in data science. And it's like having a researcher career in data science. As I say here, the work is really focused on data science itself, not so much on its application. Or there are examples, of important areas of research in data science in this paper. And after that, you can be a data scientist and use data science as a tool of research. In that case, you use the techniques of data science to advance another topic. And for instance, you would apply data science to, I don't know, data set from the OMS to predict disease, or actually in logistics to predict, the best shipping route, or stamping out tax fraud. That's a big subject now, or very profitable subjects. What's the best place to put advertisement, right? And it's close to an engineer career, let's say that. You don't do research, you do development, and you apply what is known in data science to advanced other fields, right? Now, by the way, guys, where do you sit? Where do you see yourself? Left or right? You probably don't know because you're lucky, you're very young, and everything is possible for you. In biology or in bioimage analysis, it's a bit similar again. I will take like 3 examples, but you have to forgive me, I just took examples of people I know personally, right? And so you will find mainly people from the Institut Pasteur. And in bioimage analysis, I took the example of Jean-Christophe Oliveau-Marin, who is a researcher in image processing. He works in the Institut Pasteur, right? But his field is really like applied mathematics. And the focus of his lab is really about that, like, advancing image processing. And so I took this from the description of his units. Blah, blah, blah. We develop and improve on original and rigorous methodologies for quantification of data. Our work focuses on mathematical imaging. And even in CV and career reflects that. He's actually not a biologist. He was not trained. He's very good in biology, I can tell you, but his background is really in optics and signal processing, right? It's really a field of applied mathematics. And where you see where he gets his awards and, let's say, distinctions in IEEE papers. Do you know about the main publishers in engineering and the main publishers in biology? If I tell you Nature, Cell, Science, IEEE papers, you know the difference? No is okay, yes is okay. Assaf, I guess you know. And so you see, he publishes in actually like engineering paper or in applied mathematics paper. I took an example here, denoising of microscopy image, right? It's still applying to microscopy, but review of the state of the art, a new sparsity-based method. It's a paper focusing on advancing the denoising topic. And it's published in a journal called IEEE Transaction of Image Processing, a specialized journal in applied mathematics. Okay, but Jean-Christophe works in the Institut Pasteur, and half the time you get publication like this, in Frontiers in Cellular and Infection Microbiology. So in biology, because he applies what he discovers into biology. That's one extreme, researcher in applied mathematics. And then you have the other extreme, which is Viviana Skoga. I use everything with her permission. She's a PhD student I work with. When I say I work with, I work for. And you will see in my job, I work for people. I work for Assaf, for instance. Assaf is my boss. And so Viviana, she's a PhD student in the group of Francisco Di Nunzo, and her group is called Advanced Molecular Virology and Retroviral Dynamics. And so she's a purebred. She's really, her background is really into biology, and in particular in virology. All she cares about is actually AIDS and viruses and retroviruses. She focuses on the disease. And as you see, her findings really comes from, this is the Agence Nationale de Recherche sur le SIDA and les hépatites virales. SIDA, it's AIDS, the disease, and hepatitis. So she cares about diseases, right, the virals and so on. And so this is a publication we're trying to get together, and that she studied the virus itself. It turns out that to learn something about the virus, she needs to do bioimage analysis. In that case, it's tracking and the analysis of tracks. And so her paper look like this, right? It's about images of virus, actually quantification of this, but based on the quantification of something biologic. Movies, and of course the movie doesn't play, right? This is PowerPoint after all. And you're making a model, trying to understand the fundamental processes of life. That's our focus. And in between these two guys, you will find people like me. I put them under the name bioimage analyst. That would be myself. I'm lucky enough, I'm the head of a facility, a core facility. So my job is to actually help others, typically biologists. And we are a service, which means we don't do research, we do development, but our job is really about helping others, a core facility. And so biologists will come to us and say, I need help with quantifying something in images. My CV and career also reflect that. I'm not a biologist, but I'm not a mathematician either. I come from experimental physics, and then I moved to biophysics because I found it was fun. I did a lot of microscopy, and all my career, I used image analysis as a tool. That's something I was using to advance my research, right? But it was not my research topic. As you see, I'm a research engineer, and when I publish in journals that are, for instance, called methods, right? And that's a paper when I published a tool. Interestingly, this tool doesn't contain new algorithms. I read a paper, and I simply implemented in this tool an algorithm that was described in another paper. You see what I mean? I took an algorithm and I made something concrete with that tool. Sorry for that. Right? And also other kind of paper I showed you, right? This is a paper when we do applications, when we actually make discoveries with that. And so this is how I present, that's the slide I use when I have to go out and present my facility to people. I say, hey, this is us, Pastor Image Analysis Hub. We are a facility, that's all the people you can find there, and we're all research engineers. And our mission is actually work in support, help others with the technique of bioimage analysis. Okay? And the skills we have to master, actually, I told you all along the last 1 hour and a half, it's a mix of, computing, be good with computers, algorithms, we need to be able to read the algorithmic paper, and imaging. We need to be able to connect to the biology. And so roughly speaking, the classification would be like this, from researchers in image processing to biologists, and with us in between, but with a profile of engineers. And for us, algorithmics, computing, and everything, these are tools to help others. And it's the best job in the world. That's my personal opinion. People will disagree because it's a life of service and support to others, but it's the best job in the world. You get not only to connect to exciting, actually, science done in the applied mathematics, like all the fancy stuff you can guess, but apply to cool stuff like diseases, AIDS, hepatitis, Shigella, shigellosis, developmental biology, neuroscience. We get to see a lot. We get to explore life. And I'm sure, my kids are very hard to excite, but it's very hard to excite them with that kind of paper. But when I come back and I say, hey, do you know that I found something awesome about the oil disease that it's caused by this protein? So then they listen, right? So personal opinion, my job is awesome. Sorry for that. Bon, if you want to go there as data scientist, you probably can be anywhere. You can do everything. But just keep in mind that, because if you're going to the engineer profile, you will have, whatever you do, have to learn something about these guys. Whether it's, if you work in biology, you have to learn a little bit of biology. If you work in business, like tax fraud or something, you have to learn a bit about laws and economic. And this is why I think our job as data scientists is so great, because it exposes us to a culture which is not our background. It's always exciting, and it's impossible to get bored. My only problem is that I have too much to do, and I have so little time. Bon, never mind. Sorry for this. And so this is more or less, I'm kind of into video games and role-playing games. This is more or less how I see every time I talk to a collaborator, I have these mental pictures. So the researcher would be the mage, right? He's able to create something ex nihilo, create new algorithms. And the biologist would be the warrior, the fighters. They're in front and they're making discoveries using the swords and everything. And we are the scouts as bioimaginists. We must be able, to learn about what these guys make, what they create, and see how it can be useful to these guys. Well, again, that's very blunt. Please forgive me for that. Good? No question? We can discuss it in the end if you want. Courage, folks. I'm almost done. I'll be on time. Assaf also authorized me to actually give you an opinion of what is going to be important in my field. I was uncomfortable doing that because my opinion has no relevance. I don't stand in the field of biology, right? And I'm not making discoveries in biology. And at the same time, I'm not making discoveries in applied mathematics. So what I'm going to say in the next 2 minutes, you can discard it entirely. And on top of that, I simply take 2 subjects that I feel are important. But again, the first one is deep learning. Who does not know about deep learning? Everybody knows about deep learning, right? And let's say that, you know, as a humanity, it's the scientific topic that advanced the quickest, one of the quickest in the past years. I put a very blunt chronology here. The AlexNet started in 2012, and then, paf, you have something with convolution neural networks that could win a challenge, whatever the challenge is. 6 years later, the technique they use actually surpasses human accuracy. It's relatively recent, right? Scientifically, things go fast, but this is not that far. I was born. I wasn't born for Einstein stuff, but for this I was born. In biology, actually, here I put quickly some of the important papers in biology, but again, it's very blunt. But we went fast as well. Like this UNET paper in 2015, CellPose paper 2021. The Kaggle data science ball that gives us, you know, the neural network we use a lot in biology was in 2018. And there was a lot of excitement in biology, and there's a lot of people that want to claim deep learning for themselves. So I put, you know, just 2 recent, but they're from not that recent, aimed at biologists if you're interested, you know, on the impact of deep learning in biology, but it's huge. It's really, deep learning in biology is really important, but it has specific challenges that you will not find elsewhere. Here's an example why it's so useful. In bioimage analysis, one of the most difficult topic is segmentation. Segmentation is very hard because the objects, the image we get depend really on the image quality. You see, from here to here, the image quality is not the same. And then we have touching objects, and then it's super noisy. And if you do that with classical image analysis, it's very hard having something robust, something that will work everywhere. And then comes deep learning, and then magic. It seems like to work magically. All of this, you know, comes from the same software, same network, CellPose. And it seems to work really well. It's not perfect, like, you know, you can find a small defect here and then here. But you see, given the image quality and how difficult the task is, I was able to do that like this. It's awesome. And it seems to work magically, like it's really close to magic. So this is why everyone's focused on that. It works beautifully when it is hard. So, however, we are faced with, you know, what I think are 2 main difficulties is the notion of generality, generalization, and annotation. So you all know how deep learning works? Please say yes.
Yes.
Good. Good. So, you know, there's a training phase. You need to have like some data and for instance, for segmentation, sorry. For instance, for segmentation, you need to have a training phase with a lot of data annotated. And then there's a training phase on which you produce a model. And then this model can be used to do inference within one dataset, when you take a new data and then you get new insight, right? Obviously, what you want to do is the following. You want to, because the training phase is a huge amount of work. You really need to train a lot of data. It's really painful, just this manual annotation. So what you want to do, obviously, is to say, can I reuse this model I produce in the training phase to other problems, to other datasets, right? Can I generalize this difficult work? How can we do that? Yes, we can. Even in bio, we can do that for sure in computer vision, like, you know, that's non-microscopy images, photographic images, human images. In microscopy, it's a bit more difficult. Or yes, we can. There's a lot of, you know, software tools such as Stardist and CellPose. I just put these 2 because they're my favorite, but there's plenty of others. They ship a model that you can use, you know, turnkey, and they will be very good at segmenting cells and nuclei, you know, and even what you can see here, they're very good at, you know, separating touching objects. And they work what I call turnkey. You don't have to do anything, just click the button and that works. Right, the question is, what do you do when it does not work? When you want to segment something else than nuclei or cells or more complex structures? Well, either you manually annotate a bunch of images and you can train your own network. Now the question is it a lot of work? In 2D, my colleague Dimitri Hirshov, that's our specialist in deep learning in the facility, says, 30 to 50 images with augmentation, it's not crazy difficult, it's not so much time, it's going to work. Or you can visit a model zoo. You probably heard of that, that's an emerging, it just started, I would say, where people try to actually collect network they produce and they store it on a database, on a website, and they say, okay, you can use this model if you want to segment neurons, this model if you want to segment that. It works in 2D, works in 3D, works for fluorescent image, for bright field and so on. So you can pick one. So apparently, yes, it works, but I would say also, no, it doesn't work. Generalization is a big problem. The Huffman statements are not so true in 3D. There are very few models that works in 3D, not that much at all. And the model that works in 3D, they do not generalize well. When we try to reuse them, it was failure in our case, very not something we can use in practice. And then you want to actually create your own network, right, to solve your problems. But then annotating 3D data is hell. Annotating it in 2D, it's very simple. I have a Wacom tablet, I can paint, all good. In 3D, annotating 3D data is crazy hard. And you need to be super accurate, super precise, otherwise it breaks. Okay, I just say yes, then I say no, and now I will say maybe. Because things change really quickly, right? It's a fast-moving field. And so I said there are few models, but there are actually quite a few already, right? And as we speak, I'm sure that new ones will appear. And so when I say today, just for the record, it's March 2021, I'm sure it won't be true in a couple of months. And then right now, annotating 3D data is hard, but people have produced tools to help you. And so there are tools to help you with 3D annotation to make it quicker, okay? But there's another difficulty. How good are you? The generalization, I say it's so important. Here's a paper, right? I took myself when I tried to actually track neutrophils with deep learning. And so that's a fairly specific biological question, right? It's trying to track neutrophils in specific organisms. And for that, they manually annotated 50,000 neutrophils. They say it considered it's very hard data set. But it's huge. Annotating 55,000 cells in 3D, it's a huge amount of work. But what if just to reach the scientific conclusion, you just need to annotate hundreds of them? You will never do that. You will never annotate, you will never be able to tell a biologist, please annotate 50,000 cells. If the guy tells, okay, but I can get my answer with just 100 and skipping the deep learning part, right? So this is why generalization is so important, because this work is so huge that you cannot justify doing it for only one study. For this effort to be worthwhile, we must be able to generalize it and to reuse it. I looked, you probably know also about transfer learning, right? In biology, I actually reviewed a little bit the literature, and its style transfer exists, but transfer learning not so much. Assaf, you probably have an opinion on that. Have you seen transfer learning in biology? I've seen it once. It was not super convincing.
I mean, now I think that many of the actually papers, today I'm going to show the CARE paper. So even they try to validate, well, it's not exactly transfer learning, right? It's taking models that were trained on one data set and applying them on another data set, right?
Right.
And so I think that now the top papers are, this question is asked all the time. So you are supposed to show that if you develop a tool, it should work on another data set, at least based on that, right? Taking, there were also papers that showed that not exactly again transfer learning, but training on multiple, for multiple tasks, the same network for multiple tasks actually improves your, but it relates, right? I mean, and yeah, so yeah.
Alors, right, so that is why it's super important. And just to illustrate that, you cannot, the annotation is hard because, okay, I'm sorry, I have a suicidal neighbor that actually spends his time like drilling the holes, and at some point, every one of us in the building will kill him. So if you hear that, sorry for this. Why is annotation hard? Is that it cannot be, let's say, it's a task which is very long, takes a lot of time, and it cannot be split between several people because you need to be expert. So this is a good image by a very good engineer, Samia Isham, of a bacteria called Helicobacter pylori. And if I ask you, please find me all the bacteria in this image. Can you do it? Well, actually, almost everything is a bacteria except these guys and these guys. And this one is not, this one is not, and this one is not. Right? So the annotation can only be made by people that received a good training in biology and a good training in microscopy. It's very hard to ask and to say, I'm going to have a full classroom of third grade annotate my data, because you need a good expertise in biology to be able to do that. And so, of course, if there are few people that can do it, it's expensive and it's long. And just to illustrate how expensive it is, that's the data that Stefan Saalfeld, a researcher in the Janelia Farm campus in Washington, shared with me. He organized what's called the CREAMY challenge, or you can look it up. It's a challenge for electron microscopy trying to segment the brain of adult Drosophila, small flies. And so you have electron microscopy images like that, and we want to segment neurons, dendrites, nuclei, and so on. Separately, very tension, very dense. And so he said, I have to prepare the ground truth data by manually annotating myself. And I asked him, how much does it cost? How much time did it take? It takes a huge amount of time. So there are 6 volumes in the training data, roughly 1-2 gigapixels. Roughly speaking, I think it's 4,000 by 4,000 by 4,000 roughly, right? So it's big, but not. It's really big. Let's say that, right? And with 3 categories of objects to annotate separately. And the time spent annotating the ground truth, he counted it with Jan Funke. It's about 2,000 hours. It doesn't look like much, but that's 50 weeks, right? Imagine that spending 50 weeks, like 8 hours a day, just doing that, absolutely nothing else for 1 year. Like, no holidays except 2 weeks, just that. It's crazy. It's so hard that actually they did the first pass with the company. It didn't mean that, the 1,400 hours were spent by the company. With the companies that they also spend time correcting for the mistake did by the company. At some point, he says, the company saturated precision and starting introducing new errors as they were fixing old ones. So we stopped, we quit. And that's another illustration, right? Unfortunately, the annotation, which is a very basic task, still needs to be done by an expert, which has an incredible focus on accuracy, right? We're very dedicated to the task. So it's tough. And in this case, it's an example. You can actually pinpoint the price of generating this data. $30,000 to $40,000, not even including the software to do that, right? So of course you want to generalize. Of course you want to make sure you can reuse all the efforts you make, right? So for me,
isn't it too cheap for an expert for 50 weeks?
They didn't pay an expert.
Oh.
Well, that's the problem. And I think, this price doesn't include the work. Right? It's just the money they take out of the pocket to pay for the company and everything. But I think it does not include the on time, which is astronomical, right? Bon, this is, That's the important challenge. Okay, the second one, the second opinion I wanted to share is addressing large images. I'm not going to show this slide. That's the one I said, is that now the microscope you will work with, they can generate a single image of several terabytes, no problem. It doesn't mean that everybody needs it. It doesn't mean that everybody does it, but in your career, you will meet such an image. And so there are several challenges that are important. Like,
I have a question.
Oui,
Can I ask
sorry?
a question?
Yeah.
Can I ask a question?
Yes.
Okay, so you said before that transfer learning can't be applied on biology images. So I didn't understood why.
I don't know. I actually checked the literature, how many people do transfer learning. I haven't found many reference. You want to peek into the work of Peter Horvath, who does style transfer learning. So in this paper, look at this tool, Nuclealzer paper in 2020 by Peter Horvath group, where they're doing style transfer learning to actually augment an existing data set. But it's not stricto sensu transfer learning when you take a model and then poof, generate a new one from that. They actually did that. The transfer learning directly in microscopy, I haven't seen it that much. The main reason is that this is an image.
What are all the networks that you showed before? You showed from like at least two networks name. Like what is a unit and
Ouais, alors, there are tools and there are networks. So this is CellPose, and it's basically take a network. I think it's a unit trained on many data, but they do some post-processing on that, right? And so StarDist is the same. It's a unit, and they use the network produced by the Kaggle data science ball, and they do post-processing on that, to increase the accuracy. So I mix them. In this, you have to forgive me because I have little time and this is not a course on deep learning, but it's that, right? The one thing is that the StarDist with the default network it ships is very good at segmenting nuclei. I don't have an example. Shoot. That bloboid objects that are bright over a black background. Of course, you can train StarDist on another dataset, but I'm speaking about the network that's given. Did I answer your question?
Yes, thanks.
Okay. But when it comes to big data, sorry, jumping back to big data, when you speak to a biologist, what's big data changes over time, but it's always pain. So this is the department director in Pasteur and says, big data means cold shower. You're excited, you want to do great science, and then you have to analyze this cold shower, project stops. So it's really associated with pain. And the stuff we need nowadays, I would say it's for image processing of large images. We have plenty of algorithms, but what we miss are implementation. In many of my everyday tools, there's very few of them that can harness big image, right? And for instance, you want to do Gaussian blur, the Gaussian blur of something you want to apply to 5 terabytes, you don't write the same implementation for something that works on your 100 kilobytes. And so we need new implementation for that. So algorithms, it's the same, it's just the implementation is aware that the data is huge. The next challenge associated with that is that you will find out that your microscopy data does not compress well. People have said, storing data might be a problem, so let's compress it, right? This is what we do with our video, photos, and music. In microscopy data, it doesn't work well, mainly because of the noise and the sparsity of signal, actually. And also because using lossy compression scheme in science, it doesn't bode well. And like it's touching the raw data, and the raw data is sacred. After that, there's also the problem of interacting with large images. In microscopy, like you need to see the image, you need to judge it, and you do that with your eyes. So this means that we need to produce tools that can interact with the data, when you can zoom in, zoom out, switch on something, browse in time, and it must be interactive, like more interactive than a PowerPoint, right? Offline image processing is not good enough. And then finally, there's a new something else, like if you have big data, large images, and then you segment it, you will generate a huge amount of objects. But these objects, you know, they represent their large number can be a problem too, kind of different as well. And so in that case, we need special data structures. However, don't fret, a lot of effort I've made. Right now, the people in the community, they work on this new implementation that can process large images. There are several good tools that can actually allow you to browse an image, commercial and academic. I'm thinking of Imaris, Avia, Vision 4D in the commercial one, and Big Data Viewer, for instance, in the academic world. It's good. And then the large images generate large number of annotations. That's our work with Mastodon, for instance. Actually, I will not speak about it. This is what I do when I have time. Is to generate a tracking tool, you know, that can harness, that can track cells in a gigantic data, you know, the 7 terabyte data set you see here. And so, of course, when you track all these cells over time, you generate a huge amount of cells and you still want to interact with them. It's huge work. And so with Tobias Speech, from the Max Monk, we're working since several years of making something like this, which is interactive and can harness like ideally billions of cells. And I told you I don't do research, I just do development, but just producing this tool actually required some work, some research work. Like, you know, this is an algorithm that Tobias produced. The efficient retrieval with KD3 search in convex polytopes, this is new. Tobias invented it. And this is practically what allows us to have the, to have what you see here, the interactive browsing of several millions of cells in a response time. And with this, I think I'm done. The last part would be answering your questions. I think we have a little bit of time to do that, depending on what Assaf decides. I thank you for your attention. I hope my voice was okay and that it was not too boring. I thank you very much for your question and participation. It was great. And if you have questions, I'd be more than happy to try and answer them all.
One question about the tools that used for image processing. Do you use standard tools like OpenCV or special things, special libraries?
We use special libraries. OpenCV doesn't have a lot of penetration in our field. Mainly, I've checked it, but our field is obsessed with 3D data and typically fluorescence image, at least in functional microscopy. And so the use of OpenCV is not that big. I would say maybe 5–10% of the pipeline we work with, no more. If you want to know more about the tool we use, you know, the domains nowadays is dominated by Java, Python, and MATLAB. Python for deep learning, but it emerges, you know, as more than just deep learning. Java for historical reason, because the first software used by biologists was ImageJ. And then before that, also for historical reason, because in the 90s, the only people with Macintoshes were biologists. Everybody else has PC or Linux. And MATLAB because it was a standard, you know, in the academy and in the industry. And now there's a kind of competition between Python and MATLAB because of the advent of deep learning and Python exploding in the data science, but roughly it is that. C and C++, yes, but not so much. Maybe we'll change that.
I think we'll stay in Python.
Well, the stuff, Python is really great. The one thing is that I feel that we need to have the dust settle a little bit, right? That there's, for instance, if you look for an implementation of the Laplacian of Gaussian, there's 15 of them. But if you want to find one that does everything, it's R. And then if you make a tool in Python, it's very hard to put it in the hand of the biologist. Like, you know, they use computers to do Word, Excels, and PowerPoints, not so much data science. But again, this changes fast. There are initiatives such as Napari, probably you've heard about that really democratizes the Python approach to data science for biologists, like with the viewer interactivity. And also, they also solve the difficulty of deploying, or something you will learn the hard way, is that something works on your computer and trying to make it work on someone else's computer seems easy, it's super hard. And so they work on that too. So the future, I think we will still see like Python already still increasing more.
Yeah, I agree. I agree. Cool. Any other questions, thoughts? Okay, so Jean-Yves, thank you so much. It was fantastic. And yeah. Okay, so we are done for now. We'll go out and come back at 4:30. And I'll talk about care.
I wish you the best, guys, in your career, in your studies. Good luck with
Thank
Assaf.
you.
Okay, so we had guest lectures by Nathalie and now by Jean-Yves. And now we're going to have, I think, 6 or 7, the next 6 or 7 classes are without any guests. So it's just you and me now. So the first topic that I want to start with is actually it's a good convey from what Jean-Yves showed is deep learning in microscopy. And let's start. Okay. And again, please stop me if you have any questions or comments. I'd like to hear you. Can you please open your cameras? The other people, we're not many people here, and I'd like to see your faces. Okay, so it's actually going to be today and the next 2 following weeks, I would say. And one classical application of deep learning in microscopy is doing the image analysis tasks that Jean-Yves talked about. For example, segmentation is a very common task to tackle with deep learning. I'm probably going to mention that at some point of the course, but I'm not going to focus on it now. I'm going to talk about 3 other applications. One is improving the image quality with deep learning. So taking a lower quality image and improving it. The second is, can we see something that we do not image directly? Can we take an image of some parts of a cell and see something that is, and learn something that's not imaged by deep learning? And the third is trying to understand, to classify cell states. So sick cells versus healthy cells, and the cells that are in different developmental stages or in different undergoing division or different stages going division, etc. So this is going to be the third application.
One question. You speak about processes applications that change the image itself, not taking the information from the image and run deep learning on the information, but run the deep learning on the images themselves, right? That's what you mean. That's right.
Yeah, I mean, yeah, there are also applications where, and I'll probably tell you a few examples of extracting information from the images and then doing, doing another round of learning on the feature space, right? But mostly it's going to be directly from the images, right? And the reason is I'm going to in a couple of slides I probably have it anyway, but the reason is that basically the feature basically you have an image and then you can do manual feature extraction, right? And then you can learn something, but in principle you do not need to commit for specific engineering of features. You can let the network learn the best features in order to achieve some specific task. Okay, so So this is the motivation behind that. Okay, so I shared with you guys a few resources on very basic stuff on image processing, Python programming with images, bioimage analysis, all the basic stuff. I'm not going to teach in the course. And one of them is Machine Learning 101, which is for students who are not from our department, basically, I think for people from other backgrounds who do not know machine learning, or if you want to, to remember a little bit things that you forgot, but it's very basic. I mean, basically, it's for non-computational people, I think. So I'm going to use that just for like 5 minutes, quick reminder of machine learning and deep learning. So machine learning, and this is supervised machine learning, when we have annotations of our data, so we know we have apples and oranges, and the way to now when we get a fruit and we want to classify it as an apple or an orange, the ways that we do that is we take data, we extract features. In this case, we can take the color, the texture, the taste, whatever we're interested in the fruit, and then we can train a model that based on these features know to assign an annotation, whether it's more probable to be an apple or an orange. And then the outcome of this training is a discriminative model that can now get a fruit and say whether it's an apple or an orange. Of course, if we give it a banana, it was not trained to do that, and then it can give you completely meaningless results, right? So this is supervised machine learning, and unsupervised machine learning is quite similar in the steps of taking data and extracting features which represent the data, but then we do not have annotation for the data, and the main application of unsupervised machine learning is clustering, which is basically characterizing different groups based on these characteristics, based on these quantitative properties, the features, defining different groups that your data belong to, right? So this is supervised and unsupervised machine learning. Deep learning, which could be supervised and would be unsupervised. And here you see a supervised network, which get an orange and an apple and needs to output whether it's an orange or an apple in this case, what fruit it gets, right? And the power of and deep learning have revolutionized all the machine learning fields, specifically the applications in natural language processing and image processing and computer vision. I think are the two main fields that have changed the most out of this. And the idea is basically, and the power of it stems from integrating those two tasks. So instead of engineering feature, instead of a human being defining what are the features that are important to discriminate between an apple and an orange, we let the network learn it by itself while doing the optimization process, in parallel to doing the optimization process for the actual task. So it's the integration of feature extraction and model training. Deep learning is not new. It was invented, I think, maybe in even the 1960s or so. It was, I was actually doing my master's in artificial intelligence and in genetic algorithms, and actually was around there. So people were using genetic algorithms in order to try to optimize a neural network. So it was around a long time, but it wasn't actually very helpful. And the reason was because these models are super complex in terms of the parameters, the number of parameters that you need to optimize. And there was just not enough computational power to get it to actually work and generalize and learn something meaningful. And with the emergence of modern computation, it came back to our lives in like, I think 2006 was like the first big paper, if I'm not wrong, from and then it's just boom. It's just it changed the world, and now it's basically it's dominating the field of machine learning. So how does it work? And I'm going to, again, 2 slides. I want to move forward. If you want, please read about it. I'm not going to talk about it in this course. Just some intuition. So the basic entity is the artificial neuron or the perceptron, which is a very simple function. Basically, it gets inputs, it calculates something, and then if it passes the threshold, it applies a function on it, and then it gives an output. And the output goes to some other neuron which does something similar. And when we put them one after the other, we connect these neurons to one another, we get the multi-layer perceptron, right? And basically, you get an input, you push it through the network, and you get an output here. And the way that this model is trained is by backpropagating errors. So you start with some settings, some network architecture, and some weights on the edges of this network, and then you give it a training data. You give the network training data, and then it takes the mistakes and it finds the gradients in order to improve this mistake by changing the weights on the edges. So this is the idea, and the intuition behind why it works is that it provides an increasing abstraction of the data, the different layers, right? So you start with, when you look at images, you can see that the first layers are layers that extract some very simple image, all the image processing basically filters, right? So edges and stuff like that. And when we move on to the more hidden layers, we get more and more complex and modular components of the eventual task. So I think this is a very brief introduction of neural networks. The other things that I'm going to say that there is, what I told you here is supervised. What you can also do is use unsupervised networks, and the easiest model is an autoencoder, which basically gets an input and tries to generate this as an output, and the network is built from taking the large input, compressing it, and then expanding it. And basically, the optimization is learning from the compressed representation. You try to reproduce the input image, and if you do it, and if it works, basically the code here that you see here, the latent vector here, which is represented by H, is a good compression of the data, right? Because you can take H, which is much smaller than the input, and use it to expand to a full, in this case, image, which is very similar to the input image. Okay, questions about this 5 minutes introduction to deep learning? Comments? Okay. Okay, so we're going to use it for multiple tasks. And mostly what we're going to see is actually these autoencoders. So last class, I talked about it for, I think, the last 10 minutes. So today I have a little more time to start talking about a very nice paper that is dealing with the task of improving, enhancing images. So taking images which are relatively crappy. And why crappy? Crappy because we don't put enough laser power on them, or we image them, or the exposure time is smaller because we want to image faster, or we have lower resolution and we want to improve it. And we can use that, and how to take these lower quality images and improving their quality. And it's all based in microscopy about these trade-offs. And I talked about it last time, but I think it's super important, so I want to talk about it again. You cannot get anything without compromising something else in microscopy. If you want to image a large field of view, it's going to take us time to image that, and we are going to be able to image less fast, for example. If we want to have a high signal to noise ratio, so good images which we can later process, we need to put a lot of laser power on our cells. And this is very bad for the cells. The cells can fry up and die, and we cannot do that. We cannot image them for the images that Jean-Yves showed us, that developing shrimps, right? They image the cells for a week, if you put a lot of laser power on the cells, they're not going to hold for a leaf, and the shrimps are just not going to develop. So what we need to do, we want to take the lowest laser power that we can, so we can still process the image and get what we need out of it, and we damage our cells the least, right? So all of these are trade-offs that we need to compromise with, and the idea is try to compromise less. So can we image crappier images and then computationally improve them? And this is not new, and it's not deep learning is not the first technique that was used for that. So for example, one technique is called deconvolution, and it's based on improving, enhancing images based on very specific assumptions on how the images were acquired. So Natalie showed us last week the point spread function. Remember, we want to look at the point object, but because of the optics limitations, we're going to see like a blob instead of a point, right? And so if we know what is the point strength function of the microscope, we can try to do the reverse engineering, and based on an output image which is blurry, we can go back and try to improve the resolution artificially based on assumptions on the way that we acquired our data. Now, the question is, but this doesn't take into account. Takes into account what we know and what we can be exact and measure and build definite models with. The thing is that maybe we can learn more from the data itself, right? Maybe we can learn more about all these interferences within the images that we cannot model everything in a very precise manner and an exact manner by knowing the point spread function. So maybe we can learn computationally. We can use machine learning to learn this mapping between lower quality images to higher quality images. So this is the idea of moving from something that's very steep here in this triangle, right? And trying to smear it, that we can image with less laser power, but still get high quality images, or with lower resolution and still get a high quality images. And this can give us the best of both worlds, because let's say we can image faster and still get the same performance in terms of resolution or signal to noise ratio, etc. So, CARE, which is content-aware image restoration, was developed. It was published as a preprint at the beginning of 2018, I think, and was published at the later parts of 2018 toward the end of the year. And basically, this is what it does. It maps lower quality images to higher quality images, and then once it learns this mapping, it can improve you can take lower quality images and then improve them artificially. Questions? And one thing that I like about this papers is that it's very comprehensive. So I'm actually going to show you quite a lot of data so you can see how good the work they did. So the idea is as follows. So traditional methods use general assumptions, and we want to take the knowledge on the data. So what we can do, we can generate training data by taking high resolution images, what you see here on the second. So taking high resolution paired images. So we take a high resolution image and the low resolution image, or an image that was with a massive laser power and an image with less laser power, right? And this is a matched image. And now we can use a network that maps the lower resolution image to the higher resolution, or to the original, right? To the quality image. And once we learn this mapping, once we train a model which doesn't rely on anything but the data itself, but the mapping of the data itself, we can now take less quality images and map them to higher quality images. So this is the idea. It's very simple. The machinery is a UNet. UNet is an autoencoder basically with these connections between corresponding layers. So what you see here, you get here an image. This is the low resolution image, right? And you go from a large representation to a smaller, to a more compact representation, and then you go to expand again. This is the autoencoder part. And you get those connections here that bridge the gaps between similar scales in the encoding to provide better information. And this is actually one of the only advances in neural networks, I think, that came from the biomedical imaging and then came and was very impactful in the general computer vision community. Usually it's the other way around. Computer vision community comes with the algorithmic innovations, and the biomedical informatics community takes these innovations and applies them or adjusts them in order to fit the specific problems of bioimaging. So this is the machinery, and I want to show you start with some cool results. So when we take this type of data, what you see here on the left is some type of worm, right? This is a worm, and this is a bug, whatever, some kind of a bug. This is the ground truth, right? So, oh, sorry, I need my annotation again. So at the bottom here, you see the ground truth, which is very nice and crisp. So each circle here is a nuclei of a cell. You can see it here, right? It's you can discriminate between, you can distinguish between the different cells in these images. So these are the zoom ins here, right? Here and here. This is the input image. So this is the crappy image. And the network learns mapping from here to here. And these are the results. So when taking a network that was trained to map low quality images to high quality images, we can go from this image to here. And when you look carefully, you can see that it's the results seem very nice. I mean, they seem very, very clear in terms of correctly localizing the individual nuclei. Where in standard denoising methods, you don't get as nice results as you can see here. I mean, you can see here a blur of 3 nuclei, which are here very well separated in the network image. Now, why is it so important? So here is an image, a movie. And what you can see here, this is the worms that you see here. What you can see here is that when using a high signal, so we want to see a very high quality image. So we use a lot of laser power, right? And high exposure. Basically, we're torturing ourselves. And in this animal, when you do that, what you get in the muscles, you get this kind of twitching. So the muscles are actually, the cells are actually starting to twitch. They're not feeling well, right? So you get all of this motion here. You see all of that? Now, when you take when you put the low laser power and low exposure, you get an image that is much harder to interpret. It's much harder to see here the single cells within this image. But you don't get these crazy motions of so the cells are feeling much better. And now taking these low resolution and these low quality images and using the networks that was trained to map low to high level, we can get, you see all this tweaking here on the left? And this is the healthy animal. And you can see that with these networks, we can get a really good quality image of the single nuclei that will allow us to do analysis. Okay, so here are the different, basically it's the same thing, but you can see here the ground truth, which was applied a very high laser power and very high exposure time. And now you can see going from C1 to C3, which are different imaging conditions. C3 has 1 to the 60, right? So the ground truth has 60-fold increase in laser power compared to the C3. But you can see also that the C3 is kind of crappy here when you look at the bottom here, right? So when we look at the C3 compared to the ground truth image, right? It looks really bad. Definitely much less than C1 or to the ground truth, of course. But with the network, we can get image quality, which is almost the same as the ground truth here. So basically, we can now use 60-fold less laser power, not fry ourselves on the one hand, and use the network to get image quality, which is comparable to the high image quality, okay, which is pretty cool. We always the simple idea of using this network. Okay, this is another example showing that on the different sides, and here is another one. I mean, look at this. This is the ground truth, the raw data, which is, I mean, it's really crappy. My eye cannot even say where are the exact single objects here, right? And the restoration, which gives us a pretty good image, very similar in quality to what we have in the ground truth. So it's pretty impressive how well it works. So comparing it to other restoration methods, it performs better to standard, right? To the classic restoration methods, which use assumptions based on the data, and this is great. And eventually, we are not just doing that to look at the pretty image and say, oh, this looks similar to this, but eventually, as Jean-Yves showed us, we want to, in this type of application, we want to segment cells. We want to track them over time, want to see what they do and how they move. So in order to test that, what they did is they segmented the ground truth image, they segmented the output of the network, and they segmented the original image. And you can see that the network segmented image, which is eventually what we care about, the measures that we actually care about, is very similar to what we'll get from segmenting the ground truth. So the quality of the output of the network is very similar to what we'll get from the ground truth, while if we take the raw image from the microscope with less laser power and less exposure, we're going to get really crappy segmentation. And you can see it here quantitatively. So what you see here is the ratio between the ground truth and the input. Over time, you get more and more mistakes if you want to do some tracking. So you can see that it goes from 0.95 to basically 0.5. Very quickly, you cannot do tracking anymore. These are only 30 frames, right? While with the network, you get something very similar to the ground truth, very close to 1. Okay, so this is cool. But the difficult part here is that the collection of data is really painful. Why? Because we need to match a high quality image to a low quality image. And it's not easy because you need to take image of yourself, you need to change it every time, you need to adjust your microscope, and you need to make sure that you take the exact same image, and it's a pain. You cannot do it, I mean, practically. It's just good for maybe for a paper. It's not, you cannot use it practically. And sometimes you don't even have that. So here is an example of looking at 3D images. And in 3D images, I don't remember if Jean-Yves or Nathalie talked about it, but in 3D, in most microscopes, we don't get a resolution in the Z-axis, in the third dimension, that is comparable to what we have in 2 dimensions. So basically what we get slices. Each slice has a high resolution, but between slices, we get a reduced resolution. Now there are actually microscopes that can do that in isotropic, in a non-isotropic resolution. So actually we can get a resolution which is isotropic, which is similar in Z to X and Y, but in most microscopes, we don't have that. So we lose resolution when we look at the third dimension. And a cool thing that they did here in their paper is take the resolution in XY, simulate it, how it will look when we reduce the resolution in Z. So they had a simulator of how the microscope will reduce the quality of the image if you take this to the third dimension. Learn the mapping based on this two dimension to how it would look in the third dimension, and then take images in 3D, the actual third dimension, and fill in what is missing here. And once doing that, you can get something that is much, much better. So here, if you look at the third dimension, here at the upper left corner, you see that it's, I mean, you can see in your eye, but it's really hard to distinguish where each cell is ending and where the new nuclei is starting. And when you use the network, you can actually see that much more clearly, right? So you can see very nicely where they are separated, while in the input you cannot see. Okay, so the cool trick here is using simulating data based on using data in high quality to simulate the data in low quality, which worked quite nicely. And my time is up, so I'll finish here. But what I'm going to start with, actually, I'm going to show you a few examples of that next time when we're going to start. But what would be even cooler is training networks based on simulated data completely. And I will show you that they simulated data, the microscopy data, completely simulated, no real microscopy whatsoever. And then they could go to real microscopy image and really improve it significantly. Okay, so questions at this point? Could you follow me? Patuach. A few minutes. Could you follow me, or is it too fast? Because I know I moved fast now. The question is, can you?
Yeah, we follow it. I have a question.
Okay.
So it's why do you suppose that what you have in X Y plane, you know, it describes what you have in X Z plane? Because if you like, it is always like this in cells. Because if I imagine if you look at the people in the XY plane, it's not the same in XZ plane.
Yeah, in the cells in the cells, in the cell biology world, it is you don't have you don't have sometimes you have polarity. So you have a direction, right? You have a direction that cells goes in a specific direction, but still, I mean, you do not expect to see something different locally in the pixel level. You do not expect to see different patterns that you would see in the XY.
Okay, thanks.
How basically do you get 3D images? You get like many images of the XY, different XY levels, or you get one XY, one XZ?
So I mean, the most commonly with most microscopes, what you get slices in Z, right? Mm-hmm. Like you know that probably in medical imaging, it's a lot of time like this, right? You have like slices in the third dimension, and so this is mostly what you have. There are several microscopes that actually give you a complete 3D with the same resolution in X, Y, and Z, but that's now very fancy microscopes that are available not everywhere, right? So anyway, it was a cool trick of taking these properties that you have two dimensions that are better than the third and trying to fill in the gaps, right, computationally. And this is so. I think any other questions? Okay, so I think that we're going to meet after Passover. The class is going to be probably in a hybrid class. So you can be in class if you have the green passport, and if not, you'll see me remotely. And yeah, so we'll meet in I think 3 weeks. So we'll continue from here.